Hi everyone, this is Shiny, Assistant Professor, Emmanuel Asa, College of Pharmacy. In this presentation, I am going to discuss about the transport across cell membrane, that is, cellular transport. Membrane transport is a set of processes that govern the flow of solids such as ions and small molecules through biological membrane, which are lipid bilayers with proteins embedded in. The cell membrane is made up of lipid bilayer with proteins embedded in. The semi-permeable nature of the cell membrane prevents the passage of organic anion and other intracellular proteins outside the cell. All cells have a cell membrane. The functions of cell membrane includes which controls what enters and exits the cell to maintain an integral balance called homeostasis, which controls the what to enter and what to exist. Next, it provides protection and support for the cell. Cell membrane provide protection and support for the cell. Next, structure of cell membrane contain lipid bilayer, two layers of phospholipid. So it is called as lipid bilayer. Phosphate head is polar and fatty acid tails non-polar. In the lipid bilayer, it contains phosphate head, which is polar, water-loving part, and fatty acid tail, which is non-polar, it is a water-fearing. And also the cell membrane contains proteins, which is embedded in the cell membrane. This is the diagrammatic representation of the fluid mosaic model of the cell membrane. In that, the polar head, which loves water, is shown in the outside, and the non-polar tails, which hates water, is in the inside. This is the phospholipid bilayers. In the other image, you can see the proteins embedded in the cell membrane. Next, cell membrane have pores, that is holes in it, which is selectively permeable, which allows some molecules in and keeps other molecules out. The structure helps it to be selective. Cell membrane have some pores. That pores is selectively permeable and some may be selective. And this selectively permeable pores allows some molecule in and keeps other molecule out. This is the structure of cell membrane. It is a lipid bilayer. It contains transport proteins. This is phospholipid and it contains the carbohydrate chains, proteins, and this is the cell membrane. Next, types of cellular transport. Passive transport, active transport. In passive transport, cells does not use energy. There are three mechanisms: diffusion mechanism, facilitated diffusion mechanism, and osmosis. In active transport, cell does use energy and three types of mechanisms are protein pumps, endocytosis, and exocytosis. Next, passive transport. In passive transport, cell uses no energy. Molecules move randomly. Molecules spread out from an area of high concentration to the area of low concentration. This passive diffusion is characterized by transport of substances along their concentration gradient and electrical gradient. This type of diffusion is not dependent on energy. For example, diffusion of lipid soluble molecules like oxygen and carbon dioxide and water across the cell membrane. This is the passive diffusion mechanism. Next, facilitative diffusion. Diffusion with the help of transport protein. And the osmosis is the diffusion of water. The diffusion is the random movement of particle from an area of high concentration to an area of low concentration. It is characterized by transport of substances along the concentration gradient and electrical gradient. Diffusion continues until all molecules are evenly spaced, that is, equilibrium is reached. Diffusion continues until all molecules are evenly spaced. Molecules will still move around but stay spreaded out. Next, facilitated diffusion. It is a diffusion of specific particles through transport proteins. It is found in the membrane. It is characterized by transport of substances along their concentration gradient or electrical gradient. Carrier proteins present in the cell membrane helps in this type of transport, and it is not depend on the energy. This passive diffusion is depend on the energy. Passive diffusion also not depend on energy, and this facilitated diffusion is also not dependent on the energy. The example for the facilitated diffusion is to presence of glucose transport protein helps in the transport of glucose in RBCs and muscle cells. These transport proteins are specific. They select only certain molecules to cross the membrane. The carrier proteins or transport proteins are specific. They select only certain molecules to cross the membrane. Transports larger or charged molecules. This fas- these carrier proteins in the facilitated diffusion transport the larger or charged molecules. In this animated images, we can notice that facilitated diffusion is taken place with the help of the channel proteins or carrier proteins, and the diffusion, normal diffusion, is taken place without the help of the proteins. Next, in the cellular transport, from a higher concentration to the lower concentration, the molecules is transported. The glucose molecule is transported with the help of the transport protein from the higher concentration to the lower concentration across the cell membrane. Next, passive transport. Osmosis. Osmosis is the diffusion of water through a selectively permeable membrane. Water moves from high to low concentration. Water moves freely through pores, so solid to large to move across. So, in the osmosis, diffusion of water through the selectively permeable membrane. Next, active transport. In this cells uses energy, actively moves molecules to where they are needed. Movement from an area of low concentration to an area of high concentration. There are three types: primarily active transport, secondly active transport, and protein pumps. These protein pumps transfer proteins that require energy to do work. Example, sodium potassium pumps are important in nerve responses. Protein changes shape to move molecule. This requires energy. The active transport requires energy. Protein changes shape to move molecules. This requires energy. These protein pumps transport proteins that requires energy to do the work. Sodium potassium pumps are important in this nerve responses. So this primary active transport, it is a primary active transport mechanism. This transport involves the transport of substances against the concentration gradient by utilizing the energy obtained by the hydrolysis of adenosine triphosphate. Example for this primary active transport is sodium potassium pumps, ATPase pump. It is most significant example of active transport in the human body. This pump exchange potassium by transporting sodium out of the cell. Therefore, the concentration of sodium within the cell is maintained at low level and that of potassium at high level. Next, endocytosis. Endocytosis, this is taking bulky material into a cell, uses energy, cell membrane and fold around food particle. Endocytosis is the reverse of exocytosis. It includes phagocytosis, pinocytosis, and receptor mediated endocytosis. Phagocytosis means in this process, foreign substances like bacteria, dead tissue, are surrounded by the plasma membrane of cell to form phagosome. These phagosomes fuse with lysosome. These phagosomes fuse with lysosome and hydrolytic enzyme of the lysosome digest the foreign material inside. It is also known as cellular eating. So it is phagocytosis is otherwise known as cellular eating. Neutrophils and lymphocytes in the blood and tissue macrophages are responsible for the process of phagocytosis. Next, pinocytosis. In this process, liquid substances are engulfed by forming vesicles similar to that of in the phagocytosis. Yet the vesicles formed here are smaller than those formed in the process of cellular eating. So, the vesicles formed here are smaller than those formed in the process of phagocytosis. Pinocytosis is referred as cellular drinking. The next one is receptor mediated endocytosis. In this process, materials are engulfed only bind to specific receptor protein present on the surface of the cell membrane. Later, the entire complex is digested by the cell. Example for the receptor mediated endocytosis are transport of iron and vitamin into the cell, uptake of low density lipoprotein cholesterol by liver, and uptake of neurotransmitter by postsynaptic cells. Next, exocytosis forces material out of the cell in the bulk and membrane surrounding the material fuses with cell membrane. Cell changes shape and it requires energy. Examples are hormones or wastes released from the cell. Exocytosis is the process of by which contents of the cell and release outside the cell. It occurs via following mechanism. Biosynthesis in the cells result in the formation of certain products which are enclosed in membrane vesicle. Initially, fusion of this vesicle with cell membrane occurs. Later, at the site of fusion, they break down to release the content outside the cell. This mechanism is known as exocytosis. Adenosine triphosphate and calcium ions are generally required for the process. Effects of osmosis on life. Osmosis is the diffusion of water through the selectively permeable membrane. Water is so small and there is so much of it, the cell can't control its movement through the cell membrane. What is hypertonic solution? The solution have lower concentration of solute and higher concentration of water than inside the cell. So the low solute and high water. Lower concentration of solute and higher concentration of water than inside the cell. Result is water moves from the solution to inside the cell. Cell swells and bursts open. That is called as cystolysis. In the hypertonic solution, have the lower concentration of solute and higher concentration of water. inside water than the inside the cell. And the result, water moves from the solution to inside the cell. Cell swells and bursts opens. Next, hypertonic solution is the solution of higher concentration of solute and lower concentration of water than inside the cell. This is the opposite of hypertonic solution. The result, water moves from inside the cell into the solution. Cell shrinks, that is plasmolysis. Next, isotonic solution. Isotonic is the concentration of solute in the solution is equal to the concentration of solute inside the cells. The result is water moves equally in both directions and the cell remains same size. That is dynamic equilibrium. What type of solution are these cells in? Animal cells are in the hypertonic solution. Animal cells, this image shows that animal cell and plant cell in the hypertonic solution, and animal cell and plant cell in the isotonic solution, and animal cell and plant cell in the hypotonic solution. How the organism deal with the osmotic pressure. Bacteria and plants have cell walls that prevents them from over expanding. In plants, the pressure exerted on the cell wall is called Turgor pressure. The protist like paramecium has contractile vacuoles that collect water flowing in and pump it out to prevent them from over expanding. Saltwater fish pump salt out of their specialized gills so they do not degrade. Animal cells are bathed in blood. Kidneys keep the blood isotonic by removing excess salt and water. Thank you.